Amanda Toland, and I'm going to tell you about our review article, Genome-wide Association Studies and Polygenic Risk Score Studies for Skin Cancer. Clinically useful yet? This was published with myself, Dr. Roberts, and Dr. Asghari. As a way of introduction, over 50 different susceptibility loci containing genetic risk variants for melanoma, basal cell carcinoma, and cutaneous squamous cell carcinoma have been identified in genome-wide association studies. These loci and variants only confer small amounts of risk. Thus, polygenic risk scores using variants identified from these genome-wide association studies have also been developed for these cancers and investigated with respect to clinical risk prediction. The reasons why we did this study are 1, to provide an overview of the current population-based genetic predictors for melanoma, cutaneous SCC, and basal cell carcinoma. 2, to summarize genome-wide association findings for these 3 cancers. And 3, to highlight potential clinical utility of polygenic risk scores to existing prediction models for skin cancer. To do this study, we identified genome-wide association studies, polygenic risk scores, and population attributable risk studies using PubMed and the National Human Genome Research Institute European Bioinformatics Institute catalog. We used a number of search terms. We found 21 genome-wide association studies, 11 melanoma, 3 SCC, 7 BCC, and 11 polygenic risk score studies. Importantly, we found that multiple loci are important for more than one type of skin cancer. For example, 6 loci in pigment-related genes overlap between the 3 cancers. As pigment factors are a known risk factor, this makes a lot of sense. From our polygenic risk score studies for melanoma, we found that the odds ratios increased from 1.23 to 3.22 in the highest risk group, and these were from models containing 11 to 204 SNVs, or single nucleotide variants. We also found that when you incorporate these polygenic risk score studies into existing clinical prediction models, including factors such as sex, age, pigmentation, that it improves the predictive value with a higher area under the curve, ranging from 0.62 to 0.81. We also found similar increase in predictive value when you incorporate polygenic risk score studies for non-melanoma skin cancers with existing clinical risk factors. This study is important because we found that risk prediction models based on demographic, lifestyle, and clinical variables result in varying degrees of discriminatory power, and by adding the polygenic risk score studies to these, it can modestly improve predictive value. Furthermore, polygenic risk scores could potentially identify individuals at both ends of the risk spectrum, which could have implications for screening